Hello. All right. Next question is from Mark. Oh, yeah. And I should point out that this is again our pretend Monday one that we're actually recording on Sunday. So we've got a lemur shirt on, as always, wardrobe by Nicola, but this time haircut by Nicola as well. Wow. Yeah. Thank goodness we have Nicola. All right. Question is. So thank you so much for answering so many great questions. Well, thanks. Yeah. Can you comment on a paper that was published out of Mount Sinai in the journal Cell, called "Imbalanced Host Response to SARS-CoV-2 Drives Development of COVID-19"? Yeah, I had seen that paper, and boy, I looked at that thing and thought, boy, there's a ton of work here. So what they've got are patient samples, and they've got some stuff that they're doing in cell culture and some stuff that's actual patients. And it is lung samples from two people that died of COVID and two people that did not, were uninfected, healthy donors in comparison. And what they're looking at is, what are these cells making? What are they doing? And it's the answer is a lot, and it's kind of weird. So the first thing that jumped out at me was right there in figure 1A of this paper, if you want to read along. So when they took these cells, they pulled out all of the RNA that has a thing called a poly-A tail. So the poly-A tail, the only thing that's there for in this case is to mark what's called a messenger RNA. And so the cell should be absolutely full of messenger RNAs. Now, it just so happens that SARS coronavirus 2, like all other coronaviruses, builds itself in a way that looks just like a messenger RNA. So it has the same thing at the front called a cap, and it has the same thing at the end called a poly-A tail, which is just like A. I guess if you said it out loud, it would say essentially. And so these are things that make it look and work like a messenger RNA, which just means you can turn it on and make protein from it, essentially. Yeah. All right. So anyway, but yeah, you know this. Probably most of you know this. And when they looked inside infected cell culture cells, so these are different kinds of cancer cells that you can infect with the virus, they found that about 10% of the total poly-A RNA in there, so 10% of the messages inside these cells were from the virus. They were parts of the virus that were being copied. And that is a very high number. We've seen numbers like this in a couple of other studies where they will take, for example, ocean creatures and pull out all kinds of crazy viruses. Some of these things were reporting 10, 20, even 50% of the poly-A RNA, of the message RNA, was from the virus. And frankly, I thought, no, that's a little high. It's probably like 1% in a really bad case, but no. Within the field, we've known for some years that coronaviruses have different ways. We don't understand them fully, but different ways to shut down parts of the host cell's ability to make messages or get the messages read, or in some way. And so some parts of the virus will go into the nucleus to perhaps interfere there, and other parts of the virus will be floating around outside the nucleus and doing things. And there are a couple different ways, a couple different proteins, including NSP1 is just the name of one of these proteins, that seem to be able to do something to get rid of a lot of host messages. And so effectively, they're stopping the host cell, like the one that the virus is in right now, from being able to call for help or being able to respond and, do things to shut down the infection. Yeah. So that's terrible, but those are just in cancer cells. And so then you scroll down, and the next really big one for me is figure 4. That's the last figure in the main part of the paper. And this is the one comparing from the two people that died to the two people that survived, and it's looking at levels, like how much of particular things were being made. And so let's see, from what I recall, interleukin 6, IL-6, was being made in just vast abundance. So this is something that we know sort of starts off a normal inflammatory response, but in coronavirus, it just keeps on going. It's supposed to just come up, go down, go away. Yeah, it's done its job, but it just keeps rising, and never stops by the look of it. And that's weird. That's quite unusual. The other thing they report in here is that type 1 and type 3 interferons. So type 1, our example would be called interferon beta, and type 3, our example would be called interferon lambda, if I'm remembering that right. Yeah, I think that's right. And they look for the amounts of those that were being made in these samples from extremely diseased and totally normal lung. And the answer was none. There was none of either being made. It's been shown in other systems and by other people that in at least some people you get some level of both of these things. And we know that both of these things can knock the virus back. Actually, down the supplemental information on this paper, which is like all the extra stuff that didn't quite fit into the main, headline part of the paper, but it's still there. So if you need a deep dive, you got a nice big pool to swim in. And right there in one of those figures, they actually test it out, and sure enough, it knocks back the spike protein because interferon does work. Now, the complication here is that when people have tried interferon therapy, they tried specifically giving kinds of interferon beta. And from graphs like the ones in this paper and a bunch of other papers, we know that interferon beta at least can work in some situations. But for whatever reason, the interferon beta treatments in people did not seem to really do much, which was very interesting. Now, the other thing that jumped out at me from that figure 4 was that the amount of interleukin-1 beta, which is a really strong, it's like pulling all the fire alarms at once, kind of. It sets off a big interferon response, inflammatory response, as I understand it, yeah. My limited, tiny immunological brain. Yeah, just pathetic. So this was just absolutely the same between the people who were sick with COVID and the people that were totally fine. And that's weird because normally it is interleukin-1 beta that drives this kind of response, this kind of overreaction, but not in this case. And the other thing that was weird is that a blocker for interleukin-1 beta was extremely high in the people who had died from COVID and was not being turned on at all in the other people. So not only is it not being made, it's actually actively being blocked. And so this may be a response from the body. So the body is, some various cells are going to be involved here, and we don't have a clear picture as to which ones are controlling which stage of this. But you've got different cells that are observing that there's basically an inappropriate immune response. That's how I would think of it, which is you're doing too much of the wrong thing and not enough of the right thing. And so they are sending out messages saying, stop doing the wrong thing. And I think that may be what we're seeing here, like the sort of end result of that. It's such a black box. There's so many steps involved that we just can't see, and you can just look at the outcome and try and infer back. But immunology is so hard. Yeah, at least for me, always was. And they're saying, based on this, we can define COVID-19, actual disease, as a disease where you have really high interleukin-6. You don't particularly have any interleukin-1 beta or any other kind of interleukin-1 being turned on. It seems to be actively blocked. And where you have very little type 1 and type 3 interferon. So those 4 things seem to mark COVID-19 as a little different from other illnesses. And they look at some influenza viruses and some other viruses in there as just a way to provoke a certain kind of immune response. And it's quite interesting. I'm sure there is a bundle more detail that I did not gather on my first reading. I'm going to have to go back to this one and do my homework. But very interesting paper. Thank you for bringing it to the group. It at least points to, if we can find a way to stop interleukin-6, or if we can find a way to stop whatever is causing interleukin-6 to be made, then those look like reasonable approaches to be able to slow down this virus. I believe there are interleukin-6 blockers being tested. I have not seen any particularly positive results from those yet. So it's one of these other things that seems like it ought to work and may have problems because of the way we're doing it. Like if you could actually definitely do the thing that you're trying to do, it might really work. But trying to do it with an antibody, which is the only way that we have that's approved to do it, it just may not work that particular way. So I don't know. We'll see. But yeah, thank you very much. Very good paper. Yeah, this has been Ask Dr.